This lesson we're going to take a look at carbohydrates. As the name implies, it's carbon with waters, hydrates. So the only elements present in carbohydrates are carbons, hydrogens, and oxygens. You will also sometimes see carbohydrates referred to as saccharides. That's coming from the Greek word for sugar, because carbohydrates, when broken down, are sweet sugars. You'll also be able to recognize the molecules because most of them end with OSE or OS, right? Glucose, fructose, lactose, maltose. Those are all examples of carbohydrates. So when we start to look at carbohydrates, we can break them into kind of three major classifications. The first being monosaccharides, or our simple sugars. So mono meaning that there is one monomer. It's not a chain, it's not a polymer, there is one single entity. So when we look at our monosaccharides, their role is a really quick energy source. So glucose, ribose, fructose. Now they can have various structures, and we'll talk about that a little bit later, but the idea is they are one monomer. Now, when we talked about functional groups earlier, we'd mentioned that carbohydrates can have ketones or aldehydes or either of the carbonyl groups within them. You'll see both of those in all of these types of carbohydrates. The next classification are disaccharides or oligosaccharides. Di implying that you have two monomers bound together, oligo meaning several. So that can range usually between about 3 to 6 or 7, but it's still a relatively short chain. These molecules are also used for energy sources in organisms. They include examples such as maltose, sucrose, lactose that we see coming from our milk. Now, when we're talking about maltose, that's two glucose molecules put together. When we're talking about when we're talking about sucrose, that is glucose and fructose. So glucose and a 5-sided ring put together. So just two put together. Lactose is also a disaccharide. Polysaccharides, poly meaning many. So they are used for energy storage. They're not the quick source of energy, but it's where organisms tend to store their energy. They're good for structural support, cell-to-cell communication. These examples include starches, glycogen, cellulose in plants, and chitin, or sometimes said as chitin, in certain types of like lobsters or animals with outer or exterior shells. So I want you to stop right now. Don't copy anything down on this slide. I will go through it step by step in the future slide. I just want you to pay attention to the details that are going on here in this slide, not copying down the diagrams. However, I would like you to copy down the general formula of carbohydrates. We usually have a ratio of 1 to 2 to 1 of carbons, hydrogens, and oxygens. So let's take a look at the different structures that carbohydrates can have. They can have linear structures, and as we mentioned before, we can see our functional groups here. So since we have a double bound oxygen and a hydrogen, we know that's an aldehyde. We also see hydroxyl groups as well. Now, when they're dry, oftentimes they're in the linear formula, but when carbohydrates are usually in water, they take on a ringed formula. So one of the hydroxyls bonds with the aldehyde, and you end up with this ringed structure, which is what you're going to see more often. So an abbreviated ring structure leaves out those carbons, remembering that every time you see a bend, that's meaning or indicating that there is a carbon at that point. Okay. Well, there are two major types of glucose that we're going to be looking at. Now, you look at them, they look pretty similar. They have the exact same formula, but structurally, on this very first carbon, it's the positioning of the hydroxyl group. In the alpha glucose, the hydroxyl group is down. In the beta, it is pointing up. Noticing that we start labeling our carbons on the right, and we proceed around through the molecule as long as that chain continues. So what I would like you to be able to draw is glucose. So follow these instructions to do so. Let's draw a molecule of alpha glucose. So we start by drawing an oxygen. We then add a hexagon shape. We're then going to add in the other carbon that is not in the ring. This carbon is up top, okay? In order to satisfy its outer ring and its bonding capacities, it is bonded to 2 hydrogens and a hydroxyl group. We're now going to add in the remaining hydroxyl groups. For an alpha glucose, which we are drawing now, we follow the down, down, up, down rule. So we go down, down, up, down. Those represent the location of our hydroxyl groups, so we go ahead and fill those in now. We then have to fill in the remaining bonds. For the carbons, as we see at each corner, needs 4 bonds. So we fill in the rest of the bonds with hydrogens. And there you have it. That's alpha glucose. So the example that I just outlined for you was alpha glucose. You need to be able to draw both alpha and beta glucose. And the difference is the location of the hydroxyl groups. So in alpha glucose, we can call it a dud. Those hydroxyls starting from carbon number 1 on the right go down, down, up, down, dud. Beta glucose, however, is up, down, up, down. So as you're drawing, after you've drawn your ring structure and you're positioning the hydroxyl groups for a beta, you draw up, then down, up, then down. Alpha and beta glucose are isomers of each other. They have the same formula, but they have different structure. Why is this important? Because they react differently because of their different structure. A famous example of this is the example of thalidomide. It was used as a morning sickness pill for pregnant women in the 1970s. The effective molecule looked like this. However, oftentimes when women would take it, it would interact and it would isomerize. So all that happened was this part of the molecule flipped, it shifted. However, causing serious birth defects. Children were born without their limbs, so they would have hands and feet, but no arms and leg. So just a critical example to remind you of the importance of isomers, that structure really does dictate their function and what they're capable of doing. So how do we go from a monomer to a disaccharide? Well, first thing you do if you're drawing it, you draw 2 glucose molecules side by side. You would then pick the hydroxyl group of one and the hydrogen of another. Doesn't matter which one you pick from each one. Those molecules will interact to form water. Remembering this is an anabolic or a condensation or dehydration synthesis reaction. So what is then left over is you have this oxygen that's left and needs something to bind to. You have a carbon that still needs a bond. So that carbon and oxygen will bind, and you see that down here. Okay? This is called a glycosidic linkage. When you have 2 glucoses bond together, you end up with a disaccharide maltose. You do need to be able to draw this, so take a moment now and make sure that you can draw this in your notes. Step 1 and step 2. Even more specifically, this is called a 1,4 glycosidic linkage, specifically because it is the carbon 1 on this molecule and a carbon 4 on this molecule that are bound together. So when we're looking at the types of bonds, when we're talking about carbohydrates, they all have glycosidic linkages. But depending on where the hydroxyls are positioned, we can have different types of glycosidic linkages. When the hydroxyls are both pointed downwards, as in the case of this glucose and fructose, we end up with an alpha glycosidic linkage. Because both of them are pointing down, you end up with a downward pointing bond. However, if you have a molecule like galactose and glucose, you will end up with a bond, because if you look at this, hydroxyl is pointing up and this one is pointing down. These are 3-dimensional molecules. So you end up with what is called a beta glycosidic linkage, because the bond goes up and then down on the other side. You do not need to be able to draw this example. However, you do need to be able to recognize beta glycosidic linkages, or recognize if you had an alpha and a beta glucose together, this is the type of bond that would be formed. So what are some examples of polymers? One of the most common polymers, is actually one of the most abundant substances on this earth, is cellulose. So if we look at cellulose as repeating monomers of glucose, and we notice that the bonds are going up and down and up and down, meaning that they are beta glycosidic linkages, because they are alpha and beta glucoses that are put together. Cellulose's main function is structural support in living things. Interestingly enough, we as humans cannot break down cellulose and use it for energy, because we don't have the enzymes or the molecules that can break those beta glycosidic linkages. So cellulose for us, that's our fiber, that's our roughage. When you eat cellulose, or when you eat celery or corn, sometimes notice the corn kind of goes through your whole system. That's because we can't break down those cellulose molecules. Another example is starch. Now notice here, it's repeating monomers of glucose, but these are all alpha linkages. So our body has the enzymes to break these down and can do so. So starch is mainly a storage molecule for us. However, we can break those linkages and use the smaller units for energy. Glycogen. looks almost the same as starch. Actually, in this picture, it is identical. The difference between starch and glycogen is that glycogen becomes highly branched. It's not just a single chain, but it will actually have glucose and polymer chains coming off in all different directions, as you see down here. So glycogen is also used for storage, but it is a very highly branched molecule. Chitin looks very similar to the others, noticing it has alpha and beta glycosidic linkages. The difference is there are nitrogens present as well. Chitin is used in the exoskeleton of many types of crustaceans, such as lobster. It is mainly a structural carbohydrate, and it gives them that structure, that skeleton, that allows for the protection.